Hello and welcome, everyone. I think we should start. My name is Pauline Davies, and I handle outreach for the Centre for the Convergence of Physical Science and Cancer Biology. And I'd like to introduce tonight's speaker, Paul Davies, who is Principal Investigator in the Centre. Paul is a theoretical physicist, cosmologist, and astrobiologist. His work has mainly focused on applying quantum field theory to black holes and to the origin of the universe. He also has a major research program in the origin of life and was an early champion of the idea that life on Earth may have originated on Mars. His work on quantum effects in the early universe is widely cited as an explanation for the famous temperature fluctuations seen in the cosmic microwave background radiation, the fading afterglow of the Big Bang. These fluctuations are the tiny variations thought to be responsible for generating the large-scale structure of the universe, such as galaxies. Paul has written about 30 books on topics as diverse as time travel, the search for alien civilizations, and the existence of multiple universes. And he has an asteroid named after him in recognition of his work on cosmic impacts. Paul is Regents Professor and Director of ASU's Beyond Center for Fundamental Concepts in Science. He is also a physicist, and he was recently made a visiting professor of bioengineering at Imperial College in London. Now, why, you might wonder, would a theoretical physicist be working on cancer? Well, I'm going to let Paul answer that. But first of all, can you turn off your cell phones? Thank you. Paul.
Thank you, Pauline, and have you turned off your cell phone? No? There we go. So, 42 years ago, President Richard Nixon famously declared war on cancer. With the stroke of a pen, he enacted the National Cancer Act and brought into being the National Cancer Institute. And he said, in upbeat tone, that the time has come in America, elsewhere in the world too, when the same kind of concentrated effort that split the atom and took man to the moon should be turned toward conquering this dread disease. And he confidently predicted that within just a few years, cancer would be largely brought under control as a public health issue. Well, bringing into being the National Cancer Institute meant ascribing a budget, and today that budget has grown to about $5 billion per year. And for comparison, NASA's space exploration program spends a little less than $5 billion per year. And then in addition, charities spend about $2.2 billion per year. So if you add all this up over the lifetime of the war on cancer, the United States taxpayers have paid something like $100 billion on research. And in addition to, well, I used to think $100 billion was a lot of money, but it probably wouldn't buy you a bank bailout today, to put it in comparison. In addition to the cost of research is, of course, the cost of treatment. And here it is widely recognized that cancer drug regimes can be enormously expensive. And I'm grateful to George Post for providing me with this slide, giving some of the numbers, some of the dollar numbers associated with a year's treatment of these various types of cancer. And you'll see that some of these are in excess of $100,000. And because the incidence of cancer is rising all the time, that this bill for cancer treatment is growing and growing remorselessly. So what are we getting for all of this money being spent? Well, here are the latest figures provided to me by the National Cancer Institute. This is the death rates by cause with some comparisons. So we have here heart disease, cerebrovascular diseases, pneumonia, influenza, and then cancer. And you'll see that these major killers have largely been brought under control over the last few decades. But cancer is a little bit of improvement, but it remains stubbornly resistant to treatment. Here's another way of displaying the same facts. This compares heart disease. You see the improvement over these years, with cancer slightly improving in recent years, but frankly not much to get the pulse racing. It's a very mixed picture. Now, it's often said that if things go on as they are, about half the men in this room will die of cancer and a third of the women. And so if you take a look at cancer death rates, what you see is that they were climbing and they've fallen a bit more for males and females. What this conceals is a very interesting fact. A lot of this downward trend is because of the anti-smoking campaign. That is, that it finally has sunk in that cigarettes give you cancer, and it turns out that men have been more responsive than women to that message. And so some of that improvement is not due to treatment, it's due to this anti-smoking. So this all adds up to a rather dismal picture. Decades of very high expenditure on research, escalating bill for treatment, and not a dramatic effect on the statistics. The sense that something is not going right is only compounded by the many news reports of the following sort. Researchers say vast majority of preclinical cancer studies not reproducible. And to quote the article, at least 90% of preclinical cancer studies published in science journals are not reproducible and therefore wrong. Now, one can debate that and cut the statistics this way and that, but I think that there is no doubt that a very large amount of the money that is being spent on cancer research is not being productively spent. There's, in my view, far too much money spent chasing far too few ideas. So, why is one of the most intensively studied phenomena in biology so poorly understood? It's been estimated that there are in excess of 1 million published papers in cancer research. So this is a biological phenomenon that has been under the most intense scrutiny for decades, and yet it's not properly understood. Well, perhaps the answer is we've been thinking about it the wrong way. Maybe the way to make progress on cancer is to rethink the problem from the bottom up. And it's in that spirit that I was brought into this Center for Physical Science and Oncology. And it was a result of a telephone call about 7 years ago from this lady here, Anna Barker, who at the time was Deputy Director of the National Cancer Institute. She now works here at ASU in a different capacity. The gist of the call, and she's this time last year stood in this very place and gave the same story, so I think we agree on this, that she said, well, you don't know me, but I'm Deputy Director of the National Cancer Institute, and we're spending billions of dollars every year on cancer research, and we don't seem to be making a lot of progress, and can physicists help, you see? So what was I to say? As a physicist, I said, well, I'm sure, of course, obviously we can help. But I don't know anything about cancer. I know nothing about cancer. She said, it doesn't matter, because the whole idea was to bring in people from outside to take a fresh look. So in the early days, I used to say that my main advantage for doing cancer research is that I'm unencumbered by any knowledge of the subject. Since then, I've learned a bit. I've had to learn at least the words as part of making a credible effort in this exercise. But you'll notice that under my name on the title side, I had never done an autopsy. And that was an accusation leveled at me recently, because with what authority do I stand here and tell you about cancer? Well, it's the authority of a fresh pair of eyes looking at a problem that does seem to be famously intractable. Anyway, to cut a long story short, as a result of this this initiative, 12 centers of physical science and oncology were created around the country. Here they are, and here we are. And I'm principal, as Pauline said, I'm principal investigator of our particular, this stands for Physical Science Oncology Center. And who are we? Well, this is what we've got. We are coupled with the Mayo Clinic in Scottsdale and the Fred Hutchinson Cancer Research Center in Seattle. And we line up something like this. We have 6 clinical oncologists or cancer biologists, 5 physicists, 1 physical chemist, 1 astrobiologist, 3 engineers or nanotechnologists, 1 education and outreach person. You've met her already, that's Pauline, and about 15 students. It sort of goes up and down. Visit our website if you'd like to know more about it. Now, I'd be here all night if I was to explain to you the different strands of research which take place under this umbrella of physical science and oncology. We have things looking at cell morphology, we have projects looking at cell elasticity, at the way the chromosomes are organized inside the nucleus, a whole range of things, and I don't have time to go through them all. So what I'm going to tell you about is my stuff. So in addition to my being like the overseer of this Physical Science Oncology Center, I have tried to make my own contribution to the subject. And everything I'm going to tell you has been done in collaboration with Charles Lineweaver at the Australian National University, who, like me, is a theoretical physicist and cosmologist and astrobiologist who became interested in cancer, and Mark Vincent, who is a real oncologist. He's in the other London, Ontario. And so equal credit or blame, I suppose, to them. Now, a word of warning right up front. Nothing in biology is black or white. Now, let me just make that clear. I'm a theoretical physicist, and so I'm used to getting definite answers to definite questions. So earlier this afternoon, I went to a very good lecture about whether information can get out of black holes. It's a famous problem going back to Stephen Hawking's work in the mid-1970s. And it's still controversial, even after all this time. But we expect the answer to be either yes or no. We wouldn't say, well, sometimes it gets out of black holes and sometimes it doesn't. But in biology, it's like that all the time. So what I'm going to give you is my particular take on the nature of cancer. And so what I often say is that I can't, as a theoretical physicist, compete with mainstream cancer researchers on their home turf. I mean, that would be ridiculous. Just like a cancer researcher trying to do quantum field theory in curved space-time. You just wouldn't expect it. It's not appropriate. But what I can do, after a lifetime in science and a broad interest across the sciences, is survey the whole of the scientific landscape and cherry-pick things that interest me or that I regard as significant, facts that everybody accepts, but connect the dots in a novel way. And so when I say nothing is black or white, what I'm going to give you is a particular slant, which is contrarian to the usual slant. Why is it contrarian? Because that's what they're paying me to do. There's no point in putting all this money in and we rethink it and we say, well, actually, yeah, what these guys have been doing is just fine. So the whole idea is to come up with something new. I'm not going to talk about heritable cancers. About 5% have inherited genetic basis. I'm going to talk about so-called sporadic cancers, the 95%. And the other is that there's nothing new under the sun. There's 1 million published research papers. It would be astonishing if everything I said couldn't be found in a paper out there somewhere. So in a sense, what I'm telling you isn't new, but it's packaged perhaps in a novel way. Now, what does a theoretical physicist do when confronted with the problem of cancer as a major scientific mystery? Well, physicists always like to boil things down to their essentials. They ask the basic questions. I direct the Beyond Center for Fundamental Concepts in Science, so I think in terms of the fundamentals. What are the fundamental questions we can ask about cancer? What is it? What is cancer? Why does it exist at all? And what is its place in the great story of life on Earth? These are questions that very few cancer researchers bother with. They think they have a disease to be cured, they have patients who are dying, and they need to get on with the job of finding a cure. But my job, I think, is to stand back and ask, what is this thing called cancer, and why is it there at all? And the take-home message is going to be that cancer is a window on the past. And I'm going to explain what I mean by that. But before I do that, we've all heard of cancer. Every family is touched by cancer in one way or another, so you'll all know what we're talking about here. But some people may not know precisely how it develops or how it happens. So let me just give you a broad brush summary. It's actually very simple. The basic story of cancer is this: cells start to proliferate in a particular organ in the body uncontrollably, and then they spread around the body and colonize other organs, and that's called metastasis. And 90%, maybe 95% of fatalities from cancer are caused by the metastatic process. That is the dissemination of the disease around the body and not by the primary tumor. So this is a very distinctive and peculiar thing to do. Why should a group of cells in the liver decide that they're going to go and make a home in the lung? What a bizarre thing to do. And yet it's so much a familiar feature of cancer biology that people rarely stop to say, well, that's a really weird thing to happen. But that's what happens. The cancer spreads around the body. Of course, you'll hear that all cancers are different, very complex disease. Lots of cancers of different organs will develop in different ways. And all that's true. And it's back to the black or white. What a physician may see as a bottle half empty, I see as a bottle half full. They see the complexities because they're dealing at the sharp end with human beings. I look for the simplicities. So what they see as a complex disease, I see as a very systematic disease. So here's a picture of this metastatic process. Here's the primary tumor. You probably can't read this little writing at the back. That's the primary tumor. It proliferates. It organizes its own blood supply. And then eventually some of the cells in the tumor get fed up and they disconnect and they go off, often like in a wagon train all together, and they invade the bloodstream. That's called intravasation. Sometimes they spread through the lymphatic system, but if they get into the bloodstream, then they're swept around the body in a raging torrent. And then, here we are, pumped around the body through the lungs. And then there's this other process called extravasation. You have to learn all these words, you see, if you're coming to the subject. Intravasation, extravasation. What does that mean? That the cells these are cancer cells circulating in the bloodstream. It's a complicated process, a lot of interesting physics. They sort of roll along the inside of the blood vessels when they get rather narrow, and then they attach themselves to the side, and then they sort of batter their way through, and then they get into an organ somewhere else, and they colonize it. So that's the proliferation process, and then those secondary tumors grow, and sometimes you can have tertiary tumors and so on. There's a lot of other stuff here. The cancer cells recruit healthy cells to their evil purposes, and they will even prepare the nest, sometimes in advance. There's some evidence that organs around the body are prepared to receive the colonists when they come. There's all sorts of signaling, in fact, a complex signaling network around the body of command and control going on. So it's not just rogue cells running amok individually. There's an organization of this system that's called a neoplasm, so it just means new cells, a population of new cells. You have to see it as a sort of coherent whole, and that's a clue to what I'm going to tell you in a moment. So all of this adds up. Well, one little analogy I might just give here. This always goes down well in the United States. So it's a little bit like the first European settlers. They went off in their boats. They got to the east shore of America, and conditions there were horrible. And they built forts, and they hunkered down, and some of them just didn't make it. They died out, and some of them clung on. And then a few of them bribed the Indians round about and brought them to the cause. Then they reached a sort of critical mass, and then they burst out and took over the country. So it's a little bit like that. Sometimes I say it's like Viking raiders, but anyway, you get the analogy. So cancer, to me, seems like a highly organized and systematic process. It doesn't seem like these rogue cells running amok. And the significant facts are, to me, the significant facts are that cancer never seems to actually invent anything. All it does is access normal biological processes, but it's using them in abnormal ways. And cancer, it's not just one property that it will access, but a whole suite of them. So cancer involves the co-presence in the same neoplasm in the same individual of many cooperative and mutually supportive traits. Now, a famous paper written some years ago and then revisited for the 40th anniversary of the War on Cancer by Hanahan and Weinberg. This is the paper everybody cites, the so-called hallmarks of cancer. That is, what is cancer? Well, it's a neoplasm that has the following hallmarks, and there are, I think, 8 of them. I'm not going to go through them one by one, but I've already mentioned uncontrolled proliferation. There's other clever stuff, avoiding immune destruction. What have we got? Activating invasion and metastasis. I've told you about that. Creating its own blood supply, that's called angiogenesis. Resisting cell death, because you see, when cells misbehave, there's an inbuilt program that says, die. It's called apoptosis. And that's absolutely fundamental to the nature of multicellular life, which is what I'm going to tell about in a moment. So anyway, all of these things. So in a full-blown malignant neoplasm, these might all be present, co-present, all of these hallmarks. And they've all been acquired from what started out as healthy cells. But every one of those occurs somewhere else in the body for good reason. That is, that none of those things is an invention of cancer. It's already a pre-existing modality that is simply accessed by the cancer for its own ends, if I may put it that way. So to me, it doesn't look like rogue cells running amok. It really does look like an efficient, organized, systematic process. And so I'm tempted to use the word pre-programmed. It can get me into trouble, but I'm going to just stick with that. The cancer is something is pre-programmed into cells. If you like, it's an accident waiting to happen. But the central message that I'm going to try and explain to you is that cancer is the re-expression of an ancestral phenotype. And I'll explain those words in a moment. In a nutshell, it's an atavism or a throwback. So this is cancer as a window on the past. So what I'm saying is that cancer is reawakening deeply embedded ancient properties of life. And this is not a surprise to biologists. Biologists are familiar with the concept, developmental biologists are familiar with the concept of atavisms. Here's pictures of some atavisms. This is a boy with a tail, a whale with legs, supernumerary nipples. These are surprisingly common because our ancestors used to have more nipples. And what have we got here? Webbed feet. So these, these are all signatures of our past. So what these represent are ancestral features which are cryptic. That is, that they are still in our genomes, but they are normally not expressed, but they're still there. I haven't seen it myself, but I gather that your inner fish is screening at the moment on PBS, and so probably that's going to explain about atavism. Because, we do have an inner fish. Our ancestors long ago were fish. Every one of you here had an ancestor who was a fish. So let me just take you through the story of life on Earth, because that's important to what I'm going to say. I wouldn't have said Earth created, I would have said formed, but we won't get into that in this lecture. About 4.5 billion years ago. And then life on Earth, we don't exactly know when it established itself, but sometime between about 4 and 3.5 billion years ago. We certainly know it was well established by 3.5 billion years ago. But for most of the history of life on Earth, it was just microbes, just hanging out there. Like teenagers, just hang out. There's nothing much, cool. And all this went on for quite a long time until we found about 2.8 billion years ago that these bacteria and archaea are just simple cells, more complex cells called eukaryotic cells with nuclei and other bits and pieces inside them formed. And this was the first step on the road to what we regard as multicellular life. Now, let me just say a very obvious thing. Cancer is a disease of bodies. Obviously, a disease of bodies. But bodies have not always existed. And we can ask, where did bodies come from? How far back can we trace the evolutionary roots of bodies? There's not a hard answer, but we think sometime about 1.5 billion years ago, communities of cells, of eukaryotic cells, began to aggregate and share functionality and a division of labor and so on. The true mark, in my view, of multicellularity can be expressed as follows. Back here, when they were just hanging out, cells had but one imperative, which is replicate, replicate, replicate. That's all they wanted to do. Multicellular life is a completely different logic. In multicellular life, immortality, because if you just replicate forever, you're immortal. Multicellular life, the cells outsource their immortality to specialized germ cells, eggs and sperm. And the rest of the cells, the so-called somatic cells, in return for outsourcing that hard work of immortality, accepted apoptosis, or programmed cell death. So the basis of multicellular life is this division into the immortal cell line, or germ line, and the body, which dies, and individual cells in the body die when their time comes as a result of this programmed cell death, or apoptosis. That's what it is. It's an astonishing fact that biologists still don't fully understand how multicellularity emerged, because you have to ask, what is the selective advantage to an individual cell to belong to this community? And we have people working on that here as part of the Origin of Life program Pauline mentioned. Whatever that logic was, it must be pretty profound, because life has evolved multicellularity many, many times, not just once. You probably, if any of you read the books of Stephen Jay Gould, you'll probably be enthused by what is known as the Cambrian Explosion, about 600 million, well, 510 million years ago, a sudden profusion of multicellular life forms with complex body plans. But that wasn't the beginning of multicellular life. There was this long phase of hundreds of millions of years with these more modest, what Charlie Limeweaver and I call Metazoa 1.0, more modest body plans, less cell types, not so sophisticated. And then the Cambrian Explosion. And if any of you feel energetic and you're up near Calgary, I can recommend what Pauline and I did last September. We hiked the 22 kilometers to go and see the Burgess Shale, on which the Stephen Jay Gould's books are based. And it's worth going there. It was very cold when we got to the top, but it was an invigorating walk, and you get to see the fossils for yourself. So that was the Cambrian Explosion. Well, I'm digressing a bit, so let me move on. Now, one thing that probably everybody knows about cancer, and which is undeniable, is the genomic instability. That is to say that if you look at the genomes of cancer cells, they tend to have a lot of mutations, a lot of changes, sometimes wholesale rearrangements of the chromosomes, sometimes point mutations. Basically, they're a mess. And if you look at 1 million cells in a tumor, chances are you'll have 1 million different chaotic chromosomes. And so this is part of the reason people say it's very complex. Now, the standard explanation for cancer, the so-called somatic mutation theory of cancer, is that cancer is reinvented each time in the host organism. That is, a particular individual, human being say, a particular individual will develop cancer. It'll follow the same pattern, all the hallmarks and all the rest of it, but it's all reinvented anew, de novo, in each organism from a dream run of somatic mutation. That these various changes confer a suite of gains of function that make it all add up to the same thing in different people. And all this is done in a period of months or years. So that's the standard explanation, the accumulated damage that confers greater survival properties or something like that on the neoplasm. We're taking a different tack. Remember, no black or white, so that's got a germ of truth in it. But our tack is that cancer is much more preloaded. It's like an ancient subroutine. That is, it's an accident waiting to happen. It's sitting inside the cells, and it's largely a response to a threat, not a creation of the threat. There's a real difference. So what we're saying is, benzene is a carcinogen. Cigarette smoke is a carcinogen. The cigarette smoke doesn't cause the cancer. It doesn't come with a plan of inflicting a dream run of somatic mutations that produces the cancer. It represents a threat, and the cancer, in our view, is a response for that threat. So it's the same facts, but packaged somewhat differently. I'm going to tell you a bit more about it, but let me give you an analogy that might help. I think cancer is a bit like a genie in a bottle. Here it is. So long as the genie is confined, it's pretty harmless. But if something comes along and shatters the bottle, then the genie can escape and wreak havoc. And so what we've got here are two things to concentrate on. We've got the genie with its internal program, which can switch on the various properties that I've been talking about, the hallmarks of cancer, that comes able to trigger those. And then you've got the shards of glass from the shattering process. And people say, what's the cause of cancer? Well, you can say it was a bullet that smashed the bottle, or maybe it was a hammer that smashed the bottle, or maybe somebody dropped the bottle, and then people are racing around, well, these are all different causes of cancer, but could there be a pattern there? What happens if we look at these shards of glass? Might we see that they're not random, but there's a hidden pattern, some elusive pattern in there? We have the technology now to sequence staggering amounts of the genetic equivalent of shards of glass. So maybe a pattern will loom up out of petabytes of data on some massive computer database, and the answer will lie with the wreckage, you see. Well, I think that's focusing on exactly the wrong thing. It's looking at the trigger of cancer, not the cause. The cause is the genie, and the genie has very, very deep evolutionary roots, and that's the main take-home message. Now, there's another analogy that I like to use. The cancer is a little bit like this. You've all seen this. As you groan, you think, oh, no, what have I done now? You might have dropped your computer on the floor, or you may have uploaded some software that's all gone wrong, and it starts up in safe mode. So safe mode is really a sort of atavism. It's a reversion to core functionality. And that's the way I see cancer, a reversion or a throwback to life's core functionality. It runs on a basic subroutine or a basic program whilst it ensures its own survival. So that's another analogy I like to use. So now I'm tasked with having to explain to you, how is it that this extraordinary thing we call cancer, which is a sort of throwback, a reversion, a very clever deployment of all sorts of hallmarks, something that occurs again and again in different organisms, how come such a thing exists in the first place? Can we trace its roots back in time? And I'm going to invoke a famous dictum of Dobzhansky, one of the founders of the modern synthesis of biology, that nothing in biology makes sense except in the light of evolution. And so part of what we've been doing is to try to trace the deep evolutionary roots of cancer back as far as we can go. Now, understandably, most people, when you mention the word cancer, they think human disease, death, suffering. And of course it's that. But cancer is actually widespread in the multicellular world. It afflicts pretty much all mammals, fish, birds, reptiles, even in a certain sense plants, and insects it can be, and even little worms. And so it's a very pervasive thing. And so when you see something pervading life on Earth on such a broad front, then biologists immediately know it must have very deep evolutionary roots, because you suspect it's going to go way back on the tree of life to a point of common intersection. And that means hundreds of millions, if not billions, of years ago. And I first began thinking along these lines about 4 or 5 years ago when I read a paper in Nature. This is a report of the paper, which leapt out of me about the sequencing of a sponge found in Australia with a common ancestor with us of 600 million years ago, and the point being that this sponge, even this lowly entity, this lowly critter, the sponge, has tumor suppressor genes. So even sponges have to worry about tumors, and presumably did our common ancestor 600 million years ago. Our hypothesis is that that multicellularity and cancer are two sides of the same coin. So that's what got me thinking about this. Now, you might say, well, cancer is generally bad news for the host organism, maybe not for the neoplasm, but for the host organism. Evolution is, if it's been around hundreds of millions of years, why hasn't evolution eliminated it? Because it had plenty of time to get rid of this thing. why hasn't evolution removed the dangerous genie from the bottle? The answer has to be, as always in biology, well, it's got to be good for something. What is it good for? What is cancer good for? More to the point, what is the cancer subroutine or the genome? And when I say cancer subroutine, I don't just mean, in case for the biologists here, I don't just mean some sort of set of genes or a DNA sequence. What I mean is some sort of genetic network. It's got to be a much more sort of complicated thing than just find that subroutine and knock it out. It's going to be a whole switching network, which is itself probably quite complex. But the point is, it's going to be deeply entrenched, very ancient, very highly protected. And so what is it good for? Well, a number of things might be mentioned. Wound healing, because wound healing requires you to grow rather rapidly new tissue. Limb regeneration. Mike Levin, who's here somewhere, gave us a fascinating lecture earlier today about that. So these things, are good. And the immune system, these are all reasons why you might want to keep this functionality. But the one that trumps them all is this: embryogenesis. Evolution hasn't eliminated the cancer subroutine because the same genes which are active in tumors also play a critical role in the development of the embryo. You might think that this would be the number one fact that oncologists would take into account when they're doing Oncology 101, that it would be the sort of thing that has to be a really important clue. And when I talk to my many oncology colleagues, they say, yeah, we know that. But they don't seem to give it the same significance as I do. I think it's hugely significant. Been known for a long time. This paper in 1987, oncogenes in development. So it was already recognized as a list of cancer genes or oncogenes, that is, genes which are active or uploaded or overexpressed in cancer, and the role of the same genes during development. So this is something which has been recognized a long time, but I think not enough significance is given to it. However, just a couple of weeks ago, you may have seen this article in the New York Times by George Johnson, just wrote a rather nice book about cancer, tumor, the embryo's evil twin. So some people do understand that this is a really important clue, but it's one that I don't think enough fuss is made about. Now, there are people who are working on this. For example, Isaac Cohen at Harvard. I can recommend this paper in article in Scientific American and the paper. But the point is, this is the key, we identify a set of genes that are upregulated, that means switched on, in most cancers, and we show that this signature is active in early development. And furthermore, that the later stage or more malignant the form of cancer, the earlier the stage of the embryo. So as cancer develops, it seems to mimic earlier and earlier stages of the embryo. Now, how does that relate to what I was telling you about atavisms and ancient life? Well, there's something called von Bauer's laws of ontogeny, and it's a sort of rough and ready thing, and so some biologists here may squirm, but there's like everything else, a germ of truth in it. What it means is that early stage embryos tend to look rather alike. So at I don't know, 3 days or something, human embryo resembles a mouse embryo at 1 day, or something like that, and a frog embryo, and so on. And then as the embryo develops, of course, the different embryos differentiate to their more recently evolved morphologies. And so what it means is that you can sort of trace back in time to the common ancestor of these things. It's no surprise that we share an early stage embryo with morphology with a pig, because we had a common ancestor with a pig. So what we've got then is a three-way correlation. We've got cancer biology, evolutionary biology, and I've got astrobiology in there, I'll explain that in a second, and developmental biology. So we've got really three subject areas, and you might think these guys will all get together very often and compare notes. They never talk to each other. So part of the job in our PSOC is for me to run workshops where I bring people from these different communities and sit them in a room together, and we brainstorm some of these ideas, and we look for crossover, and we look for opportunities for research projects that will pool their expertise, because we're looking for the common ground. More to the point, we're not looking, and I always say this at the outset of these workshops, I say I'm less interested in what you all know in this room. I'm much more interested in what you all don't know, because that's where progress is likely to happen. So it's by bringing people together with, well, we don't understand this, and we don't understand this, and we don't understand this, and then think, well, is that where we can make progress? What's this astrobiology? That means looking for life on Mars and Europa or wherever you like it. These are people, many of whom are in this building, who study not just life as we know it, but life as we can imagine it. And it's very useful to have people who concentrate on asking the question, what is life and how did it form in the first place, when you're coming to something as basic and as deep-rooted as cancer. I've got to gallop on because of limitations of time. So the hypothesis I already said, in cancer, cells become deregulated and default back to an ancestral phenotype using ancestral genetic pathways. That's what I'm claiming. But we want to be more precise than that, because actually, this isn't terribly controversial. I may be putting it in a provocative way, but the idea that cancer is somehow a reversion, people usually say back to the single-cell world. That's been around for really quite a while. But we think more precisely, as cancer progresses in an individual, goes through its various stages, that those should correlate inversely with both the evolutionary arrow of time and the developmental arrow of time. Let me show you these arrows. Over 4 billion years, life has evolved really slowly at the beginning and then picked up pace until now. That's the long, slow, deep evolutionary story I've been telling you about. Then ontogeny, the development of the embryo, sort of recapitulates that in a very rough sense, not to be taken too seriously, but very roughly, in a period of some months. Carcinogenesis runs the arrow of evolution backwards at high speed. So we think that as cancer progresses, it's activating or revisiting or accessing earlier and earlier pathways and earlier and earlier suites of genes. And so there's a definite prediction of the theory that as cancer progresses, gene expression correlates with gene age. As far as I know, nobody has ever tried to look at cancer genes and cross-correlate them with gene ages. They may ask, do we see this gene upregulated in this or that cancer, and is there a pattern there or something? We're asking, how old are those genes? And is there a pattern in their age? And in particular, as cancer progresses, does that pattern change? And then, so that's the first thing. I'm just going to show you some evidence. And the second thing is that this is a provocative way of putting it. Cancer creates, well, it thrives in, it may not actively create, but it thrives in niches that recapitulate conditions in the Proterozoic oceans. That's, I said, we trace the evolutionary roots of cancer back about 1.5 billion years. That's the Proterozoic geological era. So the Proterozoic ocean is what we think cancer is sort of revisiting, going back to that. So this, schematically, this is a test. It's not often in biology you can test something. You don't often make predictions. You normally describe something and explain it, often with a just so story, I might say. But this is actually a prediction. We say, you can go out and look for this, do data mining, and you can actually test this hypothesis. Maybe wrong, of course. No good if you can't falsify it. It's not science. What we think is that the genes which are overexpressed in cancer, are the so-called oncogenes, overexpressed are going to be the old ones, going back at least that far. And the more recently evolved genes, which will include tumor suppressor genes, they're going to be downregulated. So these get switched off, these get switched on, and that as over time, as cancer progresses, the slope of that line ought to increase. Well, do we have any evidence at all for that? Well, we're marshalling it now. This is work in progress. This is something came in this week, a paper in Nature. I'm not going to explain it in detail, MTH1 inhibition, blah, 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 blah. This is a gene that deals with oxidative damage. And so it cleanses the pool of nucleotides which are needed to build new DNA. So that's what that gene does. And it turns out that MTH1 is known to be overexpressed in many cancers. That's an absolute candidate. Right. How old is that? 3 billion years. So you get what we're trying to do. Now, the next data I'm going to show you is probably wrong. I don't know if Adam is here. I think you are. I think I see you over there. I'm going to show this picture, maybe to your embarrassment. But what I'm going to say here, I don't think I believe the error bars, but never mind. So Adam there, a helpful undergraduate has been doing this cross-correlation of looking at the cancer genome atlas and looking at the oncogenes and looking at a database of 16,000 or so genes across, that's breast cancer expression of 16,000 genes, but cross-correlating it with a database from, it's over 1,000 species, isn't it, Adam? I can't remember the exact number, a large number of species. And then looking for binning the ages in quarter-billion-year lots. This is exactly what we would expect. Now, it may be too much to say, well, he's found it, because it looks a little bit too good, but it's exactly what we would expect, that about 1.5 billion years ago, these oncogenes would be upregulated, and then over, if you go to later and later times, these will be tumor suppressive genes downregulated. Don't take that too seriously, but it's the sort of thing we're looking for. So this is a testable hypothesis. Now, that deals with this idea as cancer progresses, it expresses ancestral genes. That's a testable thing. Next thing is, the claim is cancer likes conditions that reflect the proterozoic ocean. What do we know about the proterozoic ocean? Well, here we are. One thing that we do know about life on Earth, the Earth 1.5 billion years ago, this is about here, is that the oxygen in the atmosphere was much lower than today. There have been two great oxygenation events in the history of the Earth. When the Earth formed, I should explain, there was no free oxygen in the Earth's atmosphere. And it eventually accumulated because of photosynthesis. You've learned all this at high school, but I'll remind you. So it builds up, but of course there's lots of things oxygen reacts with. It's a dreadful, dreadful corrosive poison, and life on Earth hated it when it first arose. It had to have all sorts of defense mechanisms against it. But it remorselessly built up, first in a burst about 2 billion years ago, then it plateaued, and then probably about 700 million years ago, it shot up to the present value. So we trace the roots of cancer not to an anoxic world, but to a hypoxic world. That is a world where the oxygen levels were much lower. So we would predict, it's an intimate story between cancer and oxygen, we would predict that cancer prefers a low oxygen environment because cancer thinks, oh, we're hankering after the good old days 1.5 billion years ago. Wouldn't it be great? What was life like then? Not much oxygen around. So we would expect cancer would thrive, prefer low oxygen conditions. Lo and behold, it's been known for decades that this is true. This is Otto Warburg, who in the 1920s proposed that the prime cause of cancer is the replacement of the respiration of oxygen in normal body cells by fermentation of sugar. That is, it's an earlier or ancestral type of metabolism. So cancer cells, just very briefly, healthy cells take oxygen and they use something called oxidative phosphorylation in the mitochondria, and that makes ATP, which is like the dollar bill of energy that life on Earth uses. But fermentation is an older way of doing it, but it makes much less ATP. I think it's 2 instead of 36, or maybe it's 34 molecules per cycle. But it makes lots of biomass. So fermentation is very good for making biomass, cell proliferation, very bad at providing energy, very inefficient. But cancer does it. Healthy cells will do it as well if they have to, but healthy cells prefer to have oxygen. Cancer cells can make do, in fact, they seem to prefer, even in the presence of oxygen, prefer to do this. What's this pH down? The scientists here will know that means more acidic because a byproduct of this fermentation process, technically known as glycolysis, is the emission of an acid that makes the environment of the cancer more acidic. And that is said to give some advantage of the tumor over the healthy cells because the tumor is happy to be acidic, healthy cells feel uncomfortable. So this is one other example of, I can hardly say a prediction because, you know, this guy already said this many years ago. We didn't happen to know that when we first embarked on this, so we regard that as a bit of a success. I should mention that Otto Warburg won the Nobel Prize, I think in 1930, very, very distinguished scientist, and was nominated 3 times in his life for 3 quite separate bits of work. But he sort of fell from favor because he was Hitler's oncologist. So it's not a very good job description, I think. And so it's only in recent years, I think, that the significance of Warburg's work has been recognized once again. Now, everybody sitting here is going to say, well, this is all well and good, interesting scientific puzzle. I can see why Davis got hooked on this stuff. But, you know, is it going to do any good? How does it help in the clinic? Will it save lives? What are the therapeutic implications of this? Because if it's just business as usual, this has just been a monumental waste of time and money. Well, I don't think it is business as usual. I think there are very profound therapeutic implications, and they can be summarized in the following slide. What we think that should be done, and we're not the only ones to say this, so it's sort of obvious, is to attack the weaknesses of cancer, not its strengths. Now, I've said to you that cancer switches on various programs, and there are various hallmarks. Some of these represent a gain of function, some of them represent a loss of function. We think that what we call gain of function is actually a regain of a deep ancestral and entrenched function, which is very, very hard to combat. So if you target the strength of cancer, and its principal strength is its proliferative ability. So cells have had 4 billion years to learn how to combat insults or attacks on their proliferation. That's the one thing all life is really good at. It's almost a defining quality of life. It proliferates, it replicates. So drugs that go for that, or radiation, which attacks rapidly dividing cells, you're bound to lose in the end. You're bound to be onto a loser if you do that, because that's the one thing that life is really good at. But if instead, these healthy cancer cells, instead of attacking the strengths, attack the weaknesses. So what you're looking for is the loss of, if you believe our theory, we can identify the recently evolved specialized functionality that is jettisoned or divested or just switched off or ignored by the cancer as it evolves, and that gives you the weaknesses. So what are they? I'm just going to give you a couple of examples, 3 examples before closing. One of these I've already mentioned. The cancer loves sugar. It runs this glycolysis cycle, this fermentation. So it needs lots of glucose. So it loves sugar, hates oxygen. So what you have to do is switch off the sugar, switch up the oxygen. So this is an old idea that uses hyperbaric oxygen therapy. It's used for lots of medical conditions. It's very rarely used deliberately for cancer, but some people who buy into this whole oxidative cancer and oxygen story think this is the way to go, but you have to combine it with switching off the glucose, which means switching diet. Well, starvation is one way, just don't eat anything, but if you're going to eat something, the ketogenic diet is with lots of fat, lots of meat, no sugar, no sweet things, is the way to go. I'm not sure I know what a ketone is, but it's a chemical. You can write it down anyway. I'm not a chemist, but the ketogenic diet combined with this high oxygen. Now, is that going to work? Well, as always in this subject, you really want to know how do people get on. You can't do that because you have to do it with mice. There's an awful lot of dead mice out there in the cause of cancer research. But it's a recent paper, the ketogenic diet and hyperbaric oxygen therapy prolongs survival in mice with systemic metastatic cancer. So for what it's worth, that is a recent paper. So that's what I'm saying is that this approach to therapy conforms precisely to what we have been thinking about, which is it is attacking the weaknesses of cancer and identifying those weaknesses through this evolution story. Here's another one. These are efflux pumps. So one of the things life had to do, life, we always think of life as cells, and a cell has an inside and an outside. And cells need stuff to grow, so things have to go into the cell, but they don't want the wrong stuff. So they have to discriminate between things they want to take into the cell and things they want to pump out of the cell. So one of the very early evolved functionalities in life was efflux pumps, pumping out toxins. Maybe it's arsenic, so pump it out. But if you look at the depth on the phylogenetic tree of these various pumps, they didn't all arrive at the same time. Life didn't just invent them all in one go 3 billion years ago. They've evolved over time to deal with different threats. So as life has become more complex, has spread around, has had to face new dangers and issues, it's had to evolve pumps that deal with more recent toxins. So if we're right that as cancer progresses, it divests itself of the more recently involved functionality, then that represents a weakness, because you can then use the toxins that these pumps are designed to expel. I use the word design, but you know what I mean, evolved to expel. And they would represent a severe stress for the cancer, whereas healthy cells with that functionality intact would not be so affected. And so the philosophy here is not so much let's eradicate the cancer, let's kill every cancer cell. It's let's stress the cancer cells more than the healthy cells. Let's have a regime, a mixture of physics and chemistry, you know, oxygen and maybe some drugs, that will keep the cancer under pressure and prevent it from progressing too far. So this is a recipe not for curing cancer, not for eradicating it, but for controlling it. And that's a really important point of my take-home message. There's the third thing I want to mention, and then we're pretty much out of time. This is deliberately provocative, but it was, so one of the things that is more recently evolved, only 400 million years old, is the adaptive immune system. All cells have an innate immunity going way, way back billions of years, but this clever adaptive immunity, the basis of vaccination, the basis of recognizing invading pathogens and responding to that, is a very clever system, and that evolved about 400 million years ago. If cancer is a reversion, it should revert back to a time prior to that, which makes tumors more susceptible to disease than healthy cells. Now, it's known that tumors exist in an immunosuppressed environment. That's well established. So they create niches which screen out the immune system. They decouple from the immune system. And that's one of the reasons that the immune system doesn't get rid of the cancer. You might think it's not doing its job. Here is a dangerous cell. Why doesn't the immune system knock it off? Well, it has a go, and to a certain extent can keep it in check, but it famously fails in that quest in late-stage cancer because the tumors are immunosuppressed. But if they're immunosuppressed, then that makes them vulnerable to disease. Now, it's been known actually for thousands of years that spontaneous remission from cancer can occur after severe infections and fever. And William Coley, around about the turn of the 20th century, was puzzled by the fact that stories of post-operative remission from cancer seemed to peter out in the middle of the 19th century. And he put this down to that pesky Frenchman, Louis Pasteur, who insisted that everything was sterilized. Implements that are used for surgery would be sterilized to prevent infection. But without that infection, the cases of spontaneous remission were less. And so Coley suspected that infection might actually be used as a way of getting rid of cancer, and he deliberately infected his patients and claimed a lot of success. Now, today, it seems barbaric. You have a cancer patient and you give them, and the reason for listeria I'll explain in a moment. And of course, this sort of basically was relegated to a footnote in history, at a bit like using leeches. But we think that Coley was onto something. The standard explanation is that if you get a high fever when you have cancer, that this sort of tweaks the immune system, boosts it, and the immune system then knocks off the cancer as a byproduct. We think that at least part of the story is it's the disease that kills the cancer. And we think that this idea of deliberately infected, these days you see you can vaccinate against the disease, but still leave the tumor vulnerable. That's the basis of what we're proposing. The reason we picked listeria is because one of the things that tumors do, they recruit macrophages. So these are parts of the immune system who are designed to protect your body. These are like, the Indians who defected to the white settlers. So tumors are surrounded by these tumor-associated macrophages that are basically enacting the agenda of the cancer, and not of the host body. And listeria is a disease of macrophages that will infect that guard of honor around the tumor. And so that's what we're advocating. I put this to our colleague at the Mayo Clinic. I said, well, what would you say to a late-stage cancer patient who said, well, we've given up on chemotherapy. Will you inject me with listeria? And he said, well, he almost walked out of the room at that point. This is just not. And I said, well, what about hyperbaric oxygen therapy? Couldn't endorse it. So it's easy for me to suggest things like this. It's much harder to actually implement it. I want to sort of skip straight to the answer. I'm just going to mention, in case Cheryl Nickerson is here, she works in biodesign. I've said that I think we should forget about curing cancer, but look instead at controlling or managing it. And so this is the field often known as epigenetics. And Cheryl's interesting because she sends bacteria into orbit to see how they get on in microgravity up there. And their gene expression changes. So the point is that even the effect of the Earth's gravity can cause changes in gene expression. And there's a whole lot of other things as well, not just gravity, tissue architecture, hardness of surroundings, shear stresses, electric fields, heard about that earlier today, oxygen tension, all these things can affect gene expression. And I must just show, because Mike Levin is here, these are his slides, I'm grateful to him, about how changing the electric membrane potential in these little worms called planaria can be used to make them grow 2 heads or 2 tails. And the point being that these are genetically identical, but their phenotypes are very different. And so the story I'm trying to tell is that cancer is more about the phenotype than the genotype. It's more about gene expression, not so much about gene content. It's about genes, if you will. It's another one. Cancer creates embryos. If you take a nucleus from a cancer cell and implant it in an enucleated egg, then you can grow a normal, at least for part of the time, a normal-looking embryo from that. So what this is saying is that a normal cellular environment or a normal tissue environment can normalize cancer cells even when they're a genomic mess. And I'll also cite the work of Mina Bissell. We're collaborating with her, who waxes lyrical about the importance of healthy tissue architecture. So cancer cells in a healthy body basically don't progress very far. They're held in check by the healthy tissues themselves. So the body does it, in this case, not through the immune system, but just by healthy tissues. So it doesn't mean just because you have cancer cells in your body that this is a death sentence. Basically, the tissue microenvironment has an effect of holding it in check. And so this opens the possibility of being able to manipulate, this is just saying what I just said, being able to manipulate a number of physical parameters like oxygen tension I mentioned, pressure, temperature, electric potentials, pH, maybe a number of other things. That the way I see it is that when cancer is lurking in the body, maybe it's dormant over many years, that there's some range of these parameters that will hold it in check. And that if those parameters make an excursion outside of that range, then it will tend to burst out and spread. So that this holds out the hope of getting away from chemical toxins to cure the cancer and getting to a point where we can use some of these physical parameters, because these are things that are not generally harmful. These physical parameters to control the cancer, not eliminate it, not kill it, but control it. If we understood how the cancer phenotype depends on the microenvironmental parameters of that sort. So here's the conclusion slide. Cancer is not a modern disease, but a deeply embedded part of multicellular life with ancient evolutionary roots. It can be triggered in many random ways, but once initiated, it follows a pretty deterministic trajectory. That is, it executes a program. Cancer is mostly a response to a stressed microenvironment, and its genomic instability is a consequence, not a cause. Cancer is a rerun of the embryo development and a rerun of evolution, but in reverse and speeded up. And the cancer grade, as it progresses, correlates inversely with developmental stages and with evolutionary branch points. We can test that. And the cancer can be delayed and managed. This is not a recipe for a cure, but it's a recipe for keeping the cancer at bay for long enough. I was going to end there, but I just want to mention one other thing. I'm going to finish by telling a little story. In 1980, I went to the University of Newcastle upon Tyne as the head of the theoretical physics department. And shortly after that, I had a letter from a 16-year-old girl called Emma Bakes, who said that she'd read some of my books. She thought it was really thrilling to study physics. Could she come to Newcastle University and study physics in my department? She did, and she got her degree, and she went to London and got a PhD, and then she moved to the United States. She was in Princeton. Then she ended up at NASA Ames. That's their astrobiology lab in California. And I used to see her there from time to time, and she worked on molecules in space and so on. And then about 3 or 4 years ago, she sent me an email message saying that I noticed you're coming to give a lecture at Kepler's bookstore in Menlo Park in connection with your book launch, The Eerie Silence. And she said, yeah, I'm going to come along, and so it will be great to see you. And as you know, I gave up astrobiology a few years ago, and I enrolled at Stanford Medical School to become a physician. And I'm due to qualify in a few months, and my partner's great, and my little boy is great, and it'll be really good to see you. And by the way, it's probably the last time I will see you because I've been diagnosed with late-stage cancer, and I think I've only got a few months to live. Well, you can imagine what a heart-rending meeting that was. It happened to be my birthday as well. And so after the talk in the bookshop, we sat and talked about cancer and life and astrobiology and goodness me. And then, sure enough, in the tried and tested manner, she battled on to the end, and she died the following February. That was April, so she lived for about another 10 months. And so you'll probably have gained the impression from me this evening that I regard cancer as a really fascinating scientific challenge, full of all sorts of wonderful things to think about and to collaborate on, and that it is. But we must never, ever lose sight of the fact that it is a human tragedy of extraordinary proportions. And so I'd just like to finish, I can go back, with a slide of Emma, and she's sorely missed. She died at the age of 42 and left a 6-year-old boy. So for no other reason than for the Emmas of this world, we have to do something, make better progress against this disease called cancer. Ladies and gentlemen, thank you very much. We're happy to take questions. I think Pauline is going to moderate this, and then we have microphones going around. We won't go on for too long, because I think I overran by 5 minutes.
Okay, well, we have a lady there, just down here, Vanessa.
So what I've always heard about cancer, I happen to be in remission for leukemia myself right now, but what I've always heard about cancer is that the way it's created is that it's just a random RNA or DNA will sequence a strand incorrectly for a cell, and it will lose its ability to tell when to stop recreating, and then will just spread around the body.
Right.
So is what I always thought I knew about the cancer wrong, or parts of it correct? Or
Well, remember my caution that in biology that things are not right or wrong. It's all shades of grey. And it's true that the standard explanation for cancer is an accumulation of genetic damage that leads to uncontrolled proliferation. It's undeniable that there is genetic chaos among cells, but it's all a question of whether what is taking place here, and let me make a distinction, we have, but it's a very rough division, oncogenes, which are, people say, the drivers of cancer. We have tumor suppressor genes, which are suppressing the cancer. Random damage to a tumor suppressor gene can just disable it and unleash the cancer that way. And so to that extent, it can be random. But the point is that the cancer program, we are arguing, is not a randomly created thing. It's a deeply embedded, highly protected, ancient set of modalities, biological modalities. It's there for a reason, one of those reasons being its role in embryogenesis. That's what we're saying. But in terms of the triggers, you must distinguish what shattered the bottle and how did the genie arise in the first place. Many things could shatter the bottle. There are many pathways to cancer, but once cancer is triggered, it follows a rather deterministic trajectory. That's because it's deeply embedded, very ancient, very highly protected, and rather predictable. I'm not sure any of this is of much help in the clinic, but my point of view is that if we fully understand the phenomenon, then we're better able to tackle it.
Right. I think we've got a man over here.
Allow me two things. One, to comment on whether the current fad movement towards antioxidant foods, rich foods, is likely to increase, sort of go against the smoking ban trend. But secondly, and more interestingly to me, you've said it several times that there's almost like I see it as a cassette that you think can be fired off and evolve and go backwards evolutionarily. And I'm wondering whether you would speculate on like where this is located. What is this thing? Where is this cassette? Where is this ancient program sitting in the genome or whatever else? And is it a sequential thing, fixed? Right. You know, A leads to B leads to C leads to D in all prostate cancers or mammary cancers or whatever.
So I did actually address that in passing. I said, don't think of this as just, you know, a segment of DNA. There it is. That's the cancer subroutine. Chop it out or something. What I said was it's going to be a network, a gene regulatory network, because genes don't just sit there and get read out. They switch each other on and off in complex patterns. So you should think of it more like circuitry. So rather than talking about a cassette of genes, I would rather think of it as like a module in a TV set or in a computer or something. It will have some sort of functionality, but there'll be a lot of internal information processing going on. So it's something like that that we look for. And you might say, well, if we could identify it, couldn't we just knock it out? But we come back to the fact that this is there for a reason, that the functionality which cancer is appropriating, everything cancer does, your body needs to do. So, for example, extravasation. The cells get out of the bloodstream, colonize another organ. What are they doing there? They're doing exactly what macrophages do as part of the immune wounding response. Cancer is often called the wound that never heals. So if you knock it all out, then you lose your wound healing capability. So you know, you've got to really nuance this. The oxygen story is an interesting one, yes, because we're told antioxidants are good for preventing cancer, and that's true. So oxidative damage is one reason that you get mutations, and some of these mutations, if it's in a tumor suppressor gene, could lead to cancer. But I think Jim Watson has pointed out recently that if you've got cancer, what it needs is really anti-antioxidants. It needs to go the other way. So it's a bit of a mixed message there. I think it's going to be a complicated story, but the connection between oxidative damage and cancer is a very important one, and we had 2 workshops on that because I perceive it, and I think many other people do, as absolutely key to understanding this condition and learning how to control it.
Pauline, we have a question.
Okay.
I have a question on what do you think about the women getting tested and then having their breast removed? How does that play into your studies or what you know about that?
Very political, and I can answer it because Pauline said, before you go to this lecture, just check what the National Breast Cancer Coalition, NBCC, who I'm going to talk to in a couple of weeks, what they have to say about it. And you can look at their website. They come out rather strongly against mammography, against testing. There's an explosive issue, very political, but Pauline did run a debate with an advocate for mammography and against 2 or 3 years ago, because this is a difficult subject. But you're asking me my own personal opinion, and so what I say is this: that if there's a family risk of breast cancer, or an elevated risk for any reason, then screening makes a lot of sense. The problem is I can explain exactly what the problem is. That if you have blanket screening, and if a woman is told, well, we have detected a small lesion in your right breast, and you can be told 75% chance it will never be a problem, but there's a 25% chance it might be. And so many women then opt for double mastectomies in response to that, for something that probably would never be a problem. And so what we would like, of course, is to be able to say, well, we've sequenced your genome, and in your case, we can assure you it's never going to be a problem. Leave it alone. Or the other way around. That's called a biosignature. The field of biosignatures is in total disarray. We simply do not have those biosignatures. So it's not possible to say that when you detect these things, that yes, you're going to be in trouble and you're not. We can't divide it up in that simple way. And as testing technology gets better and better, small lesions are detected earlier and earlier, and more people are faced with this predicament. So one recommendation, and I'm not saying I'm endorsing it, but the NBCC says that on the whole, mammography, there are other forms of screening, but mammography does more harm than good. Mammographers obviously don't agree with that. So that's a controversial statement. I'm simply saying what they say on their website. So you must go there. And obviously these things at the end of the day are very much individual decisions. So I had a conversation with John Matic recently. He was at our house. He runs the Garvan Institute in Sydney about screening and testing. And I said, what a test for Alzheimer's? Why would I ever want to know? I'm at an elevated risk for Alzheimer's, unless something can be done about it. He said, well, I really want to know. And I said, well, I would rather not know. So it boils down to personal choice, I think. Anyway, very, very difficult political issue.
No, here.
I think you've proven very well the role of the physicist in connecting the dots in a very complicated problem. Your last slide here especially prompts the question about clinical application. With $100 billion and 1 million papers in the literature, it's hard to imagine that every conceivable experiment has not already been done. What is the role of data mining and connecting dots and the physicist to help know where to look in the data mining?
Right, a very good question. You can do data mining without, a license, without an ethics committee, and all the rest of it. And so that's what we're doing. But you might think, well, what about clinical trials? And I showed the hyperbaric oxygen therapy combined with the ketogenic diet. So when we had our last workshop with Tom Seafried, who's been an advocate of this for some years, he was asked by one of the program officers from the NCI, why don't you do a clinical trial and then convince us? And Seafried's answer was, who's going to pay for that? The NIH won't pay for it. They regard this as, too far on the edge. And you see, a clinical trial is, in this country, immensely expensive. $100 million or something to run a clinical trial. And the point is that unless there's a drug company with a product that's going to get that money back, nobody's going to pay for it. And this isn't a drug, it's oxygen and diet. Well, who's going to pay for that? Who's going to make money out of oxygen and a diet? And so you see the way in practice how difficult it is to translate any of these ideas into the clinic. Now, maybe in other countries, it's not so difficult. You see this with aspirin. The best anti-cancer drug that we know is, on a general, I mean, there's some specific ones, but generally, is aspirin. And in clinical trials in Europe, it's been shown, claimed it can cut incidence of major cancers by up to 40%. So it's a preventative. Take a baby aspirin a day for 5 years or 10 years or whatever the trial lasted. It is a clinical trial because you give people placebo. So they did a clinical trial. We don't hear a lot about that because you can buy it over the counter for a few dollars. So, I'm sounding very cynical, but not nearly as cynical as some of my oncology colleagues. And so we have to change the culture of the way we approach this, as well as the nature of the research itself. Because these ideas on the edge simply can't be properly tested, and they need to be, otherwise they're valueless. Right, the aspirin is clear. The aspirin has been established. But that's more of a preventative measure. And so the question is, if you're diagnosed with cancer, what about these alternative ideas that I've been talking about? I'm not going to claim that ketogenic diet plus hyperbaric oxygen is going to do it for everybody. But what I think is I can understand why it might, because I think I understand cancer in terms of its deep evolutionary history. And therefore, if I understand why something works, I'm more likely to believe that it will work. It's not just a matter of, well, let's give this a try or that a try, see what works. There's a good reason why it should work.
Right, so let's take this young lad here.
Ah, my most long-standing fan.
Yes, he always asks intelligent questions.
Right. Well, this is obviously what we want to know. How can these ideas be used in the clinic to improve patient outcomes? Because that's what it's really all about. Some of the things I talked about, physicians will be very comfortable giving the advice. So, for example, I've said that healthy tissues can keep cancer in check, even if you have cancer cells, even small lesions in various organs, that if the tissues are healthy, the cancer won't progress. So keeping healthy, diet and exercise, is always a good thing to do, and that you'll always be told that. But in terms of, can we take things like this hyperbaric oxygen therapy and do that? Well, people are free to do it, but physicians generally will not endorse it for the simple reason that if you have a particular hospital or a particular medical practice, for them to actually endorse a particular treatment option, if it's not standard of care, then that exposes them to all sorts of risks and lawsuits and things like that. So obviously, it's a free country. You can ask for something to be done, but that's very different from it being a recommendation or an endorsement. And that's the problem we're battling with. But this, I make out that that's bad. Of course, it's good as well, because if doctors would just prescribe any old thing that the patient said, well, I've read about this and I want to try that, it would be total chaos. We have to have some quality control. So in science, I've always learned that any really good idea in science has to be battle tested against the skeptical community. You have to have a rough ride. You come up with an idea, hyperbaric oxygen therapy, and people have got to say, don't believe it, show us the evidence. It's got to be, you've got to have a skeptical community, and only then you get past that point should it be acceptable. So the mark of a good theory is one that has been battle tested against a skeptical community. I've seen it in physics, I've seen it in cosmology, and I see it in biology as well. Not everything I've been complaining about is actually bad, but it does serve to slow the progress for these alternative ideas, I think.
Let's just have one more question. Shall we have one from anyone at the back?
Oh, yes, he's...
yeah.
yeah.
Okay.
Thanks for giving me the chance to ask the questions.
Only one, please. One question.
You have mentioned that a lot of oncogenes, the cancer-causing genes, have a very ancient evolutionary root. So what does those genes do? I mean, in those single, single cellular organisms.
Right, I gave a classic example of one earlier on this MTHFR.
So there are, in human beings, there are a lot of cancer-related gene related to apoptosis. So I'm wondering, those genes, some of them are like the cytochrome C. These genes are they're also found in unicellular organisms.
Absolutely right, yes.
So-
So we're
so-
not tracing it all back just to 1.5 billion years ago. But some of these genes, like the one I mentioned that deals with oxidative damage, oxygen first built up about nearly 3 billion years ago.
Yeah.
And so life had to accommodate this horrendous poison, and it had to learn to deal with this oxidative damage. And the gene that I mentioned, which is overexpressed in most cancers, is precisely that gene that goes right back to that unicellular world, that bacterial world, nearly 3 billion years ago, to deal with precisely that oxidative damage. So I do honestly think, because we've got a table booked for dinner, so I think I'm going to have to.
Okay.
I'm going to have to leave it at that stage. We've got to go and we've got to go and have our ketogenic dinner.
Thanks.